Right. Yeah, thanks, Pretty, for sharing, and thanks to development and the company of biologists to come up with this really cool scheme. I think it has been fantastic, and to get to know Ethan and the other six members of this year's cohort. It's been great. So today I want to, of course, talk about my postdoc work, but also give kind of a developmental spin at the end, because I'm always looking for some kind of developmental connection. So in a way, I would like to at some point give a talk that has this title rather than the other one in a long-term kind of perspective. But my kind of research always covers three domains more or less. It's always neuro, it's always evo, and it's always devo. So it's kind of a combination of those three. And what ultimately fascinates me is all of the variation we see in brain anatomy in different, for example, in this case, insect species, but also in lots of other species, of course. And as you know, of course, all of this variation emerges through hundreds of millions of years of evolution. And my central question is, what explains this variation? And I'm trying to understand the evolutionary processes, but also the developmental programs that produce these amounts of variation. Or the lab that I'm currently in is using these beautiful butterflies as a model system. So these are Heliconiini butterflies. Um, and it's a large radiation of neotropical butterflies that occur in Costa Rica and Ecuador and Panama. And we have certain amounts of outgroups, but we have this massive radiation of heliconius butterflies. And interestingly enough, all of these heliconius butterflies, except for one species, actually feed on pollen. And the outgroups do not feed on pollen. And if you think about butterflies, you may think about this proboscis, this kind of tongue, this kind of tube with which they can suck up nectar. And these kind of butterflies are, as far as we know, the only butterflies that are also able to feed on pollen by basically loading up their proboscis with these sticky pollen grains. And they then ingest this amino acid-rich resource, which increases their lifespan considerably, but it also influences their brain anatomy, because in order to feed on these pollen-containing plants, they need to perform quite a cognitively taxing task. And this is something akin to traplining or spatial foraging. So let's imagine you go for a very efficient shopping route, right? You know where all these different shops that you want to go to are, and you are super efficient going one to the other. And this is basically what those butterflies also do. And this is because these pollen-containing plants, they are quite rare in this habitat of those butterflies, but they flower really stably for up to a year. So if something is rare, but it's very stable in how they can be perceived and how they can be used, it makes sense to be able to memorize them, right? And this is what those butterflies do. And this kind of behavior puts certain cognitive demands on the circuitry, or this evolutionary process has put cognitive demands on the circuitry. And now this is basically the setup that is really interesting for us as neurobiologists to kind of study comparatively, in contrast to those butterflies that could not do this behavior. They're very closely related. One of our students, Elizabeth, has looked at the sensory periphery and the sensory brain areas, and actually they seem super conserved. So these cognitive demands seem to be not met by the sensory periphery and brain areas associated with it. But cognitive demands actually influence centers of integration. And one of these centers are the mushroom bodies, so these red blobs here. This is a brain area in the insect brain, and you can see here mushroom bodies of a non-pollen feeder of this Dryas julia species. And then this is 2-scale mushroom body of a pollen feeder. So it's up to 4 times a volumetric expansion. So absolutely massive. And this is a very shallow, short evolutionary timescale of 15 to 20 million years, which is not that much, funnily enough. And what do mushroom bodies do? Mushroom bodies are basically known to be the centers of learning and memory. And what actually happens is they take lots of sensory signals and combine them with internal information about positive or negative, so reward or punishment signals. But of course, mushroom bodies do not work in isolation. It's basically the whole brain that facilitates such a complex behavior. But today, I need to focus on the mushroom bodies, but I'm also interested in, for example, the central complex, which is the center for spatial navigation. So let's go deeper into the mushroom bodies. I said centers of learning and memory and connecting the external with the internal, right? What does that mean? So in terms of cell types, the mushroom bodies are divided in three main cell types. The first one is the canyon cells. Canyon cells are the numerically most dominant cell type of the mushroom bodies. They occur in different types, and they're actually related to a very small pool of progenitors, up to 2 or 4 neuroblasts, so stem cells originally. And they produce a lot of cells in the adult form. So, for example, in the pollen feeders, there are roughly 80,000 cells per hemisphere from only a couple of stem cells. So they're super, super interesting. And they take sensory input from visual brain areas and olfactory brain areas and so on. And they facilitate this and let it connect to other brain cell types, for example, the dopaminergic neurons. And these dopaminergic neurons basically modify the valence signals, the signal intensity of a neuron in a positive or in a negative way. And this corresponds to whether it informs the neuron to reward the sensory signal or to connect it with a positive signal or with a negative signal. And then what we end up with is a modified signal that then is projected into other brain areas. So this all sounds very complicated, but the only thing that it means is that we have a convergence of sensory information with internal information that induces a learning paradigm, basically. So coming back to this expansion that I mentioned in the beginning in pollen feeding, my main question of this paper from 1.5 years ago was, how does this expansion actually affect this internal mushroom body circuitry? And mainly in the beginning, these different canyon cell types of alpha, beta, gamma, alpha prime, beta prime, and Y. It's very complicated names. And this involved lots of immunostains with the help of Theodora. And it also involved lots of staring on these kind of confocal images for a lot of time. And this is because we needed to really make sense of this anatomy that is super, super different from Drosophila, for example, which is the main neurobiological model in insects, especially. So I had specific anatomical criteria, how to make sense of it and how to homologize which canyon cell parts corresponds to which domain in these so-called mushroom body lobes. And you can already appreciate the form looks quite different, right? And the different colors correspond to these different canyon cell types. But I wanted to, of course, get some kind of statistical insight into this. So we had a slightly expanded data set and then made general linear mixed models and other tests as well. And what we see here are the results of a general linear mixed model. So we have on the y-axis, we have the volume that these different canyon cell types occupy. And on the x-axis, basically the different types of canyon cells. And in blue are always the non-pollen feeders, in red are the pollen feeders. And you can see the pollen feeders are always higher and significantly so in terms of their canyon cell volume. So in the beginning, that was kind of a boring result, right? We have this overall mushroom body expansion going on, and the constituent parts of these mushroom bodies are also expanded. That's not particularly exciting, right? But luckily enough, if we look at a little bit deeper into specific statistical measures, you can already notice quite a lot of variation. And this actually was corroborated by something that I don't have the time to go into deeper detail today. It's called smatter analysis, and this was corroborated basically the result that we have certain canyon cells that are expanded to a much higher degree, a significantly higher degree than other canyon cell types. So we have two kind of groups. And this creates a scenario where we have an independence of specific canyon cell types from others. And this is super exciting, actually, because in brain evolution, we understand lots of results as mosaic evolution, where one part evolves independently of another. But so far, we only knew this of one brain area evolving differently from another. And now we know that even developmentally linked, highly related sister cell groups can also evolve very independently from each other. So this was the kind of the first interesting result of this paper. And I want to quickly also illustrate some of these other other cell types in the mushroom bodies that I've looked at. And this example are the GABAergic feedback neurons. Now, feedback neurons are neurons that produce a feedback signaling, right? So we have these modified signals from the Kenyan cells, and those get fed back to the Kenyan cells themselves and inhibited. So basically, we have a global inhibition that results in only having the most important information being projected to other cell types and projected outside of the mushroom bodies. So it's very important as a selection mechanism, what's most important, or called sparse coding. And these feedback neurons are another group of cells that seems to be adaptable, it seems to be changeable, because we have many more of these GABAergic feedback neurons in the pollen feeders in comparison to the non-pollen feeders. So this is an adaptable, but it's also an experimentally accessible cell population, right? This is a non-model organism. We don't have a million transgenic lines, right? We have a few antibodies that work, and then we hope for the best, basically, right? So this is a way of having a cell group that's experimentally accessible. And what we're now doing, Francesco and having produced a large single-cell RNA data set, and we're currently writing up a ginormous paper, especially this is spearheaded by him. And what we see now, and this is really cool, we can see that single-cell clustering results into lots of different Kenyan cell types in this case, right? All these different colors correspond to different Kenyan cell types. And this actually mirrors my anatomical results. So if we correlate, for example, a fold change of cell numbers from the single cell data set with the fold change of volumes that I just explained to you, we have a high correlation. So one mirrors the other, and this is quite cool, and obviously also internally confirms one to the other. And this is also true for GABAergic neurons, which is very interesting too, right? We have a general increase in GABAergic signaling and neurons in the pollen feeders than the non-pollen feeders, which corresponds to our feedback neuron result. And what I'm currently doing is having ordered 1 million HCR probes, so kind of like a fancy in situ, I like to say. And what we're trying to do is label lots of these different Kenyan cell populations and a few other cell types to kind of understand and gauge what's happening here numerically. And this is one example of a probe called MUB. So as a short summary, before I go into my kind of developmental spin at the end, we have a system where we have a very isolated behavioral variation effect, right? They are very similar behaviorally to non-pollen feeders to the pollen feeders. They just differ primarily in this pollen feeding behavior. And corresponding to this isolated behavior, we have an isolated anatomical difference in those two groups, mainly related or surrounding these mushroom body expansions. And then we have actually adaptable cell groups that change in a short time span through probably lots of different mechanisms in an otherwise very constrained and conserved background. This also means that we have testable cell groups that we then can approach through anatomy and genetics to come up with something intelligent. And all of these results that I've shown you actually fit to how insect brains develop. And this is something that I want to quickly go into now, and hope that is interesting to you as development experts. So insect brains develop through a kind of curious kind of developmental mode. Of course, they develop through a specific set of neural stem cells. They're called neuroblasts. And all the offspring that a neuroblast produces is what we call a neural lineage. But what is curious is that all these different offspring neurons stay clustered together with the somata, but also with the first tracts that they produce, which means that we have so-called lineage-associated tracts, which also means that we have an anatomical correlate that indicates developmental relatedness, right? That this bundle indicates that all of these neurons that produce this bundle are developmentally related. And this is very conserved, which I've also shown with our collaborator in a paper from a few years ago. So we have this this anatomical readout for developmental relatedness. And this is a really cool system that I'm going to try and exploit for my benefit. And why am I telling this? Because all of these results that I've shown you, and also results that I haven't had time to mention, they all make sense in terms of what their underlying developmental neural lineages, right? For example, these canyon cell types that I've explained to you that change mosaically, they are all produced by MB lineages, 2 or 4 neuroblasts, depending on which species, or the feedback neurons produced by this lineage, or other stuff by other lineages. And even my PhD thesis can be linked back to kind of this thing. So if I come back to my central question from the very beginning, what explains this variation in brain anatomy? It is, of course, tinkering with neural lineages because it's such a susceptible system to produce variation, but also obviously a system that has a lot of inherent constraints. What I want to do is I want to use these cognitive circuitries, of which I've explained a bit about, and then use the neural lineages and these anatomical correlates to gain insights into neurocircuit analysis. And I can do that now because I have a set of cell groups that are accessible across lots of different insect groups, which I can then study and find out what's going on there precisely with different methods. And with this, I want to thank Steve for being a fantastic PI. I want to thank my 3 students I had over the years that were all great. These continue as our PhD students at the moment, our collaborators, and lastly, you for your attention. Thank you.
Thank you, Max, for a wonderful talk. Yeah, we'll give our participants to ask some questions. Maybe I can start with some of them. So, I don't know, first of all, a really beautiful butterfly. I never knew that they have even this mushroom bodies, which also looks very cool. And how does like this specific cell lineage population increases in number? Like how does a very specific group of the cells increases in number? Maybe you do not know. And is it possible to test that if we reduce the number of these particular cells, you will have change in the probably size of the mushroom body and as well as the behavior of the foraging behavior of the butterflies? Is it possible to check?
So with the testing of creating a specific reduction of specific types, you would only need to be able to do this with ablation, right? I think that there's a paper from 30 years ago in cockroaches where they basically shoved a hot needle into the mushroom bodies and they couldn't perform a lot of tasks anymore. At some point, we will work on kind of having mechanisms to do that. And in terms of how they are changed, so one way, of course, is increasing the pool of neural stem cells at the very beginning. And the other one is by prolonging the phase of proliferation, right? You could increase, for example, the larval period or the period during the embryonic stages, where certain canyon cells are produced, and therefore you have a bigger pool of canyon cells, and then they can be differentiated later on. And you also have this cool temporal cascade of different transcription factors, and when they're switched on and off, they produce specific types of neurons. So this is how these different canyon cell types are produced. So for example, the gamma canyon cell types are born first and then specified by them through a specific temporal cascade. So that kind of different time points where you could produce variation.
Cool. So we have first questions. Are there any major differences within pollen feeding butterflies based on location or based on what kind of plants they feed on? Maybe based on the abundance of plants they feed on.
I will reread the question quickly. Are there any major points?
So, are there any
Yeah.
differences in the pollen feeding behavior of butterflies because of their location or because of the plants they feed on? Like, is the foraging behavior different because they are present in different environment, or they are having different kinds of food, or that's what is the
Yes,
question?
there are differences. Depends on how you define major. So the difference between these non-pollen feeders and pollen feeders are much larger than any differences between the pollen feeders. So we have other researchers in the lab that look at different species of Heliconius, and they show differences, for example, related to the altitude they are feeding on, or also, for example, how dense the canopy of the forest is that they're occupying. So there are also a lot of differences, and different Heliconius species have been studied for a long time as well. But the extent of, in terms of brain anatomy, is super large in this comparison of pollen feeding and non-pollen feeding.
So, next question is, what cells do neuroblasts derive from, and what is known about the number of neuroblasts specified?
So there's not too much known, specifically for Heliconius, or super little. But overall, there are results from grasshopper and from Drosophila that seems to indicate that there's a pool of approximately 100 neuroblasts, not of per hemisphere, so 200 neuroblasts in total in the central brain, and they seem to produce all of these different neurons, including the Kenyan cell types and including lots of other differences. And their location, even in the embryonic brain region, seems to be quite conserved. So it seems like there's a general roadmap or a general map that's produced and that's super conserved.
Okay. So, next question is, great talk. Does Heliconi ereto also have 4x the number rather than the volume of brain neurons? If so, are the number of the each neuronal types expanded equally, or has one population selected for growth?
I need to reread again. Then I also have, for instance, the number
So
of other
basically,
brain neurons.
yes, like they are asking the species like Ereto, which has 4x the number, but it is due to the increase in the number of the neurons or due to a specific type of the neurons. Is there a change in the volume or not? That is the question.
So if it's meant that specific to the Kenyan cells, then it's quite variable. Like we have some Kenyan cell types that are expanded to a higher degree, and most of the other brain neurons, like generally in the brain, are not expanded, or at least not as far as we know. Like it's very difficult showing that in population sizes because we usually don't have the antibodies, except for the ones that or a few of those that I mentioned. But lots of regions are super conserved, funnily enough. I hope that answers your question. Otherwise, shoot me another one.
So there is actually a follow-up question. What is reinforcing that a specific certain type of canyon cells override the others? I guess I'm trying to understand what would cause more proliferation of some over others. Are there downstream proteins differences also in these mushroom bodies then that feed back into getting one lineage to be more than the others?
And those are all very good questions that I can't give concrete answers to. So basically, this is something that we're looking into, right? I think the best kind of line of inquiry, so to speak, we are thinking about is this temporal cascade, because you could expand the expression of a transcription factor, the patterns, for example, the gamma Kenyan cells, and by having it expressed over a longer time, you produce more Kenyan cells more gamma Kenyan cells specifically. So I think it's this kind of the pro cascade that we need to play with, and also that we need to investigate with specific, for example, HCR markers and then other markers that would answer these questions. Are there downstream protein differences in these mushrooms? We don't have any specific proteins that we're having a look at the moment because we're starting from the very beginning. Like we look at brain anatomy, now we're trying to make sense of other information.
Okay, thank you, Max.